Funding for Yale Cancer Answers is provided by Smilo Cancer Hospital. Welcome to Yale Cancer Answers with Dr. Anish Chagpar. Yale Cancer Answers features the latest information on cancer care by welcoming oncologists and specialists who are on the forefront of the battle to fight cancer. This week, it's a conversation about research into predicting prognosis for patients with Dr. Smita Krishnaswamy. Dr. Krishnaswamy is an associate professor of genetics and of computer science at the Yale School of Medicine, where Dr. Chagpar is a professor of surgical oncology.
So, Smita, maybe we can start off by you telling us a little bit more about yourself and what it is you do.
Yeah, sure. So I'm trained as a computer scientist. So I did my PhD in computer science, actually designing algorithms for designing logic networks. And then I switched to computational biology during my postdoc at Columbia. Initially, I was looking at signaling networks and immune cells, but later when I started my faculty position at Yale, kind of opened up to looking at all different problems, particularly those that can be addressed with machine learning and the high throughput single cell data types, including single cell RNA-seq and mass cytometry, and also other things, including molecular level analysis. So currently, I would say I work on AI for biomedical science, particularly deep learning and some types of unsupervised learning.
So all of that sounds really fascinating, but a lot of it sounds like stuff that many of us might think we should know, but might not really understand. Like, what exactly is computational biology? And how exactly does deep learning work? I mean, we know that it has something to do with artificial intelligence, but how do we put all of these things together? So can you kind of break down these terms for our audience and give us a sense of what exactly you do?
Yeah, definitely. So basically, when I start out on a problem, usually there is a data set or data type. So in my recent cancer story, there were actually several different cancer organoids, quite a few actually, 3,000 or something cancer organoids that were grown from patient tumors. And each of these organoids had thousands of cells in them. And these cells were measured using a variant of mass cytometry where they're measured in 40 to 50 protein dimensions. So the main question that I would be answering, so to speak, is how to get insight from this data. So it's pretty obvious that, a human can't look through this data. So then you need a machine to look through this data to come up with insights. And we've developed methods for everything from visualizing this data, both at the cellular level and at the patient level, to trying to figure out cellular and compositional differences between patients who might respond to a drug or patients who might achieve chemo-resistance. So I would say all those buzzwords basically mean that I develop computational and mathematical techniques to make sense of a lot of very complex data.
And so just so that we understand, so basically what happened in terms of this data set was people took cells from patients and presumably some cells from normal people as well without cancer, and they grew them into what you called organoids in a petri dish. So can you explain to us what exactly an organoid is?
Yeah, so this is called a patient-derived organoid. So you would take patient tissue samples, for example, from a tumor biopsy, and as you said, you would grow it into a petri dish so you have a small version of that person's cancer upon which to conduct potential experiments. So the hope with patient-derived organoids, particularly in the labs of my collaborators, is that they form a personalized cancer experimental setup where you can actually try different drugs on this small sample of cancer and see how it responds before actually giving it to the patient. So that's kind of the idea of the patient-derived organoid. So I'm again going to emphasize that I'm not in any way an experimental expert. So I wouldn't be able to tell you about the particular details of how to build the organoids.
No, totally cool. But just so that we kind of have an idea of what an organoid is. And then you mentioned that, these organoids had, once we've developed them, they then developed all of this data using mass cytometry. Now, some of us have heard, if we think back to like introductory physical chemistry, what mass spectrometry is. But can you tell us, like, mass cytometry, it sounds like this is a technique where you kind of look at proteins that are expressed by these organoids. Is that it?
That's right. So you can think of mass cytometry as a technique that measures at the single cell level. That means you get measurements from individual cells. So you can tell how each cell is different from another cell. And what is measured are protein abundances. And these proteins can be surface markers like receptor proteins, or there can be internal cellular signaling markers. There was also single-cell RNA sequencing as part of this study, and that measures which mRNA is being produced in the cell. And if you recall, mRNA is what's then translated to tRNA and then to a protein.
And so if we think about, just the complexity of all of the proteins that cells produce and all of the mRNAs that they produce, that's a lot of data. So now we're starting to get a sense of why we need somebody with your expertise in terms of making sense of all of this data. So can you tell us a little bit more about how exactly you do that? I mean, because I can imagine that there are, thousands of data points from these samples. So how do you kind of wrap your arms around all of this data and make it make sense and put it into some sort of a potentially clinically relevant context?
Yeah, that's a good question. So first off, just to clarify what a single data set would look like, it would look like a large matrix where perhaps the rows of the matrix or like a large spreadsheet where the rows are individual cells and the columns are whatever you're measuring on the cell. If it's proteins, it can be 40 or 50 columns. If it's RNA, it can be thousands of columns. And in fact, you can have more columns than rows even because our body has 20,000 genes that it's potentially manufacturing. And to make matters a little bit more complex, most biological data is very noisy. So you can imagine that you can't quite trust the entries of this spreadsheet to begin with. That's one data set. So let me remind you that not only do we have thousands of data points, but we have thousands of data sets. So we also had 3,000 of these patient-derived organoids. So I have 3,000 spreadsheets like that containing thousands of cells each. So that's what kind of the image of the data. So what do we do to make sense of the data? So one of the things we try to do with the data is put the data into an embedding, which you can think of as a visualization or a landscape, where you can picture cells as dots, kind of like in a scatter plot. And dots that are close to each other are similar cells. Dots that are far away from each other are dissimilar cells in terms of their protein or RNA. And you can see different groupings of cells, but what's more interesting, sometimes you see different trajectories of cells. So you can see sometimes normal cells that are on a trajectory towards a very proliferative or aggressive state. And so in order to be able to make these kinds of visual landscapes, we have dimensionality reduction techniques for single cell. One of the techniques from my lab is called FATE, and it tries to learn the shape or manifold of the data, literally by sort of walking through the data and finding relationships between data points. And then in in our more recent study, we kind of lifted that same kind of analysis to the level of the data set. And when you measure a distance or dissimilarity between data sets, you use a distance that's called optimal transport distance or earth movers distance. So those are some of the techniques that are used.
So, for people who aren't really in this field, it's hard to kind of wrap your brain around how exactly you're doing this. I mean, I get the fact that you're looking at differences and similarities between these data sets with thousands and thousands of data points. But how do you kind of take this data and essentially what ends up being, this landscape of dots, and turn it into something that's clinically meaningful? Can you kind of talk a little bit more about your research and position it in a clinical context for us?
Yeah, actually, you can. So let's say that I took some of the cells from some of these data sets and mapped them out. For example, with our FATE technique, what you can do is you can characterize this landscape and you can see some corners of it have, for example, a proliferation signature, or what often happens in cancer is there's a dedifferentiation of a normal cell type back to a more stem-like cell type. And you can see such stem cell signatures. You could also see if some cells are dying, apoptosis signatures, because you can characterize the landscape in terms of the proteins and genes that are measured. So then you can begin to disentangle the cellular milieu that's within the tumor, and you can see what happens to it once you apply a treatment. Do you see a lot of cells moving towards an apoptotic state, which would be great, or do you see some cells sort of escaping into a more stem-like state, but that's quiescent and sort of resistant to chemotherapy? So you can begin to see how cells transition along this landscape and how different patients are distributed differently along such a landscape.
And so you can kind of get the sense that you could take these organoids that are growing in this petri dish and treat them with a chemotherapy, and not only see how they respond in the petri dish, but also see how this landscape, this kind of landscape of dots changes with time. Is that right?
Yeah, that's right. And actually, my lab somewhat specializes in this, in that we build special kinds of neural networks called neural ODE networks, or neural ordinary differential equation networks, that just using these static snapshot data infers how cells can continuously transition from one state to another. So in other words, you put the cellular measurement into the neural network and it actually gives you a derivative along this landscape. And you can follow these derivatives or integrate them out to follow paths that a cell might be going along.
All right. So we're starting to get the sense of how we can kind of see where a cancer cell is going using these techniques. We need to take a short break for a medical minute, but when we come back, we'll talk more about how we are using these high-tech techniques to predict prognosis for patients with cancer with my guest, Dr. Smita Krishnaswamy.
Funding for Yale Cancer Answers comes from Yale Cancer Hospital, where their cancer genetics and prevention program includes a colon cancer genetics and prevention program that provides comprehensive risk assessment, education, and screening. SmiloCancerHospital.org. The American Cancer Society estimates that nearly 150,000 people in the U.S. will be diagnosed with colorectal cancer this year alone. When detected early, colorectal cancer is easily treated and highly curable, and men and women over the age of 45 should have regular colonoscopies to screen for the disease. Patients with colorectal cancer have more hope than ever before thanks to increased access to advanced therapies and specialized care. Clinical trials are currently underway at federally designated comprehensive cancer centers, such as Yale Cancer Center and at Smita Cancer Hospital, to test innovative new treatments for colorectal cancer. Tumor gene analysis has helped improve management of colorectal cancer by identifying the patients most likely to benefit from chemotherapy and newer targeted agents, resulting in more patient-specific treatment. More information is available at yalecancercenter.org. You're listening to Connecticut Public Radio.
Welcome back to Yale Cancer Answers. This is Dr. Anees Chagpar, and I'm joined tonight by my guest, Dr. Smita Krishnaswamy. We're discussing her research into predicting prognosis for patients. And while predicting prognosis for patients sounds like a relatively easy thing to do, or at least simple, what we heard before the break is that her research is anything but simple, using large data sets of thousands of data points and thousands of cells in thousands of patients, and essentially using high-tech techniques with computational biology to map out a landscape that can kind of give us some insight into how these cells respond, for example, to different kinds of chemotherapy. So, Smita, maybe we can pick up the conversation there, and you can tell us a little bit more, perhaps even from a more granular or more practical standpoint, about some of the more recent projects that are going on in your lab and publications that are either just come out or that are on their way out that might actually impact patients.
Yeah, sure. So a study that just came out was in Cell. It was together with Chris Tape's lab at University College London. And here we had the patient-derived organoids that I mentioned. So the way we got 3,000 patient-derived organoids is by taking tumors from 12 patients and then treating them with different chemotherapies and drugs, as well as to co-culture them with fibroblasts, which are called cancer-associated fibroblasts. So in this study, we had two levels of landscaping, if you will. So we have the landscaping of cells, which shows what kinds of cells are in the population. And what we ended up doing is modeling those as being on a decision tree where cells at the leaves form different groupings of cells. And then we used those to create a landscape of the organoids themselves. So this was called trellis, and there we were able to come up with sort of a map where now each dot is a patient's tumor with a specific treatment. So then we got to see that parts of this landscape correspond to chemo-resistance, chemo-sensitivity, apoptosis, etc. So for the prognosis question, our idea is going forward to make this kind of landscape generalized in the sense that when you measure a new patient, from a tissue biopsy and make organoids out of them, that you would be able to just kind of place them into this large landscape that we've already created from trellis, and that would tell us, what might happen to the patient, sort of learning from similar cancers that other patients have had. So just to be more clear, even though all of these patients have colorectal cancer of some type, what the landscape is doing is it's kind of quantifying the heterogeneity between different patients. And so making the information about what is going to happen after treatment with particular drugs more granular. So that is one of the studies. Another study is out in preprint, and we're working towards submission. In that, we're looking at triple-negative breast cancer. And here our focus is all on learning what is the dynamic that takes cells from a primary tumor type to becoming metastatic. And metastasis is, of course, the most fatal aspect of cancer. And here we measure several different time points of mammosphere organoid, and this is like a small cancer organoid grown out of a cancer cell line. And we measure it at several different time points, and then we track that transition continuously using the neural ODE network that I mentioned before, which shows you how cells would go along from time point to time point. in continuous time, and particularly which genes are changing, how their gene regulatory program is changing. And so when you look at it from the gene level, you have the possibility of predicting new therapeutic targets. For example, here we're dealing with triple-negative breast cancer, and it doesn't have many targeted therapies at all. So that opens up the possibility that there could be new therapeutic targets from just understanding how these transitions happen.
A couple of questions on that. So as we think about this and we think about how every patient's cancer is a little bit different, we always talk on the show about personalized medicine and tailored therapies. How generalizable is this in terms of actually getting this to be something that can potentially one day be something that everybody's biopsies is actually subjected to?
Actually, my collaborators at University of College London are already working on that. So they actually have a grant from an organization called Cancer Research UK to measure 50 more patients. And their focus is on building technologies that measure these at cost. So in order to measure many patients, they're using a technique called split pool barcoding, where you can barcode each patient's cells and kind of measure them en masse. So I think these are coming. And I know in our own Yale New Haven Hospital, from a study that we did on COVID, that we're already measuring single-cell flow cytometry data on a lot of patients. So I think that these processes are becoming more and more streamlined, and they will be coming into the clinic. I think one day in the not so far future, when a new patient goes into the clinic, their biopsies will definitely be measured by single cell technology and maybe even grown into these kinds of patient-derived organoids.
And so then if that actually happens, I mean, that's really exciting, because then potentially, as you mentioned, you would be able, if I understand you correctly, not only to just take some cells, a biopsy, and test whether that particular biopsy responds to a chemotherapy or not, but have an understanding about whether there's a certain amount of heterogeneity, because you're measuring this at a single cell level, and whether some of those cells may actually be resistant to therapy, and there might be other targets. Is that kind of where you're going with this?
Yeah, exactly. So as you measure more patients, you'll gain more and more information. So each patient you can imagine as a point cloud of cells, and you begin to see commonalities between patient on which subsets of these point clouds might be recurring in patients who develop resistance to particular therapies. So you'll begin to attribute small, even rare cell populations potentially to the outcome. And then you could have therapies that target just those cells. And what's more is that these patient-derived organoids will also contain other types of cells like immune cells and fibroblasts. And you can begin to reason about what combination of these cells and what types of cell-cell communication are also leading to certain kinds of results.
And so one can imagine that for any given patient, instead of classifying them as having breast cancer or even triple-negative breast cancer, you might now get down to particular subclasses where, for your particular cancer, these and these genes are turned on, these and these proteins are expressed. This and this signature suggests that you may have a resistance caused by XYZ gene, and therefore putting all of this information together, we can whip up a concoction of therapies that are particular for your particular cancer. Is that it?
That's right. And it doesn't even have to be necessarily a concoction. It could be a sequence of therapies. So first, there might be a specific kind of therapy that drives cells into a state where they're more sensitive, and then there could be another therapy that follows on. So this is also something we're testing. We're testing how cells transition upon the application of some kind of chemotherapy and what dynamics they undergo afterwards. And I should mention that triple-negative breast cancer work is with my collaborating lab of Dr. Christine Schaefer at the Garvan Institute in Sydney, Australia. So we have a new data set that we're measuring, where we're measuring sort of the after effects or the dynamics after application of these kinds of therapy. And the other interesting point about what you mentioned is when you just talked about these gene signatures, they're non-mutational signatures. They're signatures of cells that are in a particular state. So really, patients of various different mutational backgrounds, genetically speaking, can have cells in these particular states. And it has been hypothesized by my collaborators and others that there's convergence at the cell state level, that there are certain cell states and certain mutations allow cells to reach these states a little bit easier, but there's many paths to getting there. So this is really a non-genetic cell state related, cell state based way of thinking about therapy.
Yeah, and when you talk about cell states, it sounds to me like you're kind of getting into this whole concept of, the tumor microenvironment and how a given cancer cell may behave one way in a certain environment and in a different way in a different environment. And so potentially you can get at kind of that interaction as well.
Yeah, that's definitely the case. So in our Cell study, the study that just came out in Cell, we saw that the fibroblasts, cancer-associated fibroblasts, were actually having a protective effect on the cancer cells. So there's a signaling pathway called the YAP signaling pathway that was helping the cells transition to a more revival stem cell-like state. And when that signaling was inhibited, the cells were more chemo-sensitive. So the cells, we can think of them as people in a party. They're interacting with other people and getting information and signals from them, and that's affecting how they behave. So the tumor microenvironment is very important for modeling that.
And getting back to kind of what you were talking about in terms of these large data sets, it sounds to me like this isn't just something that might be useful in terms of predicting a patient's prognosis or even predicting what therapy or sequence of therapies that might be effective, but it also might be a model to actually help researchers to find pathways or genes or mutations that they hadn't previously thought of as new targets. Can you talk a little bit more about that?
Yeah, definitely. So when we start to really look at this data in what I would call a data-driven way, so letting the data teach us rather than our prior assumptions, by using these processes that quite literally model the cellular transitions, you can start to look at all of the genes afresh to see, what are the genes that really go up first to start this kind of cascade of processes leading to this transition. So in our triple-negative breast cancer study, we identified a target called ESRRA that hadn't been reported very frequently. There had been some associations, but we actually honed in on that as a target and actually perturbed it using siRNA and drug treatment to see the effect on the transition. And indeed, it was upstream of a lot of what you would call EMT or genes that control the epithelial to mesenchymal transition in breast cancer and had a large impact on the transition itself.
Dr. Smita Krishnaswamy is an associate professor of genetics and of computer science at the Yale School of Medicine. If you have questions, the address is canceranswers@yale.edu, and past editions of the program are available in audio and written form at yalecancercenter.org. We hope you'll join us next week to learn more about the fight against cancer here on Connecticut Public Radio. Funding for Yale Cancer Answers is provided by Smilo Cancer Hospital.